What's good, babes? It's really good to very see you. You know, it's high time this channel covered one of the most important, fascinating, and fundamental aspects of biology. We're gonna dive towards some metaphorical bedrock and take a peek at the process of DNA replication. I'll cover the major steps and components to help all my sweet broodlings out there hoping to ace whatever exam styles may be coming their way. Hopefully this video helps you get a leg up on the pile, and a some lot more. Cell division is vital to any organism, but before our adorable little cells can divide, other complex processes of replication inside of them must begin. Replication can be divided into three primary stages: initiation, elongation, and termination. DNA, as we know it, has this twicely fresh double helix structure. Because of this, initiation initiates with a funky little enzyme known as helicase. For replication to become possible, the base pairs that make up the genetic code must be separated so they can be read. Helicase anchors to a section of DNA where initiator proteins have unwound the helical structure. Once established, helicase plows forward, breaking apart the hydrogen bonds linked between the correlating base pairs. The previously helical structure breaks off into a Y-shape, like a wake trailing behind a boat, creating a section known as the replication fork. To ensure that these separate strands don't get any cozy ideas, single-stranded binding, or SSB, proteins bind to the separated strands so that they don't rejoin and undo all of helicase's hard labors. SSB does more than just keep the strands separate, as it prevents the strands from being broken down or anchored onto by unwanted enzymes. The structural nature of DNA strands leads to a handful of issues when they are being tossed about, wrung out, or doing the twist and shout. A sense of physical strain comes to mind. The strands themselves do in fact have directional value, denoted by 5' and 3', making one being the leading strand and the other the lagging strand. A good term to keep in mind is anti-parallel. As the DNA backbones are parallel, their directional natures are heading in opposite directions. Leading strands going 3' to 5' and lagging strands going 5' to 3'. The easy way to recall is to think of the leading strand as going in numerical order forward. Focusing on the leading strand, good old reliable 5' to 3', DNA synthesis begins as a specific chunk of RNA appropriately called a primer anchors onto the end of the leading strand. A new face joins the crew in the form of DNA polymerase, an enzyme that binds to the primer, prepped to add new nucleotides to the appropriate bases in a new 5' 3' strand. This newborn strand of sequences is continuous as it is established in a continuous manner. The lagging strand, it follows a less simple method. With the lagging strand, you'll notice that several RNA primers start anchoring along the strand rather than the one primer on the other. Well, as you recall how DNA is anti-parallel, the strand being added to the lagging strand must be built in large chunks with small gaps to be filled in later. Though it seems oddly complicated when compared to the single continuous sequence like on the other strand, DNA can only be built in a 5' to 3' direction. This is called discontinuous replication, as Okazaki fragments are not only built as separate chunks, but gaps exist between them. The Okazaki fragments themselves are built by an enzyme called DNA ligase. Soon enough, an enzyme called exonuclease removes any anchored primers as they are replaced by the proper nucleotides. Ligase, the Okazaki-producing enzyme from earlier, links the Okazaki fragments all together. Well, there you have it, cuties, DNA replication. If you want to sound like a cool person at a party, you can also mention that DNA replication is semi-conservative, as new DNA molecule is half of the initial and half of new material.